afternoon. This is the lab video to accompany lab exercise 7, the integumentary system. So what I'll be doing in today's video is I'm going to be going quickly through the model that looks at the integumentary system, specifically the skin, which is the epidermis and the dermis and the associated structures. So this is the model, right? And you will see something very similar to this in the notes. Right? This model has three main parts to it. This part over here, this is going to represent the thick skin, right? And what we'll notice is that there's a much thicker region of the epidermis here. This middle section is going to represent the skin of the axilla. The axilla is the armpit, also the skin of the anogenital region. And then finally over here, this will be the scalp. So we're going to start off looking at the scalp, and we'll look at some of the structures that are particular to the scalp. Right? So when you think about scalp, one of the first things that you should think about is hair. Right? So what we're going to start here is we're going to see here we have the shafts of the hair, and the hair actually is going to sit within this bulb over here. Hair is very interesting. Hair is actually formed by the same cells that make up the epidermis, so it's epithelial in origin, and it results from an infolding or an in-pocketing of the epidermal cells. Right? So we have those epithelial cells that fold in, creating these hair follicles. Now you'll notice attaching to the hair follicle we have this section of muscle here. This is a little bit of smooth muscle. This muscle is called the erector pili muscle. This muscle attaches to the hair follicle, and then it attaches at the very top of the dermis to the epidermis. And when this contracts, it stands the hair up on end. Now you might wonder why standing the hair up on end is important. Well, in humans it's harder to see the importance because we have very little hair on our body. But if you think about your cat or your dog, when your cat or your dog goes outside in the cold and it comes back in, it looks puffier. Its hair is all standing up. The act of standing the hair up straight actually helps to trap air in between the hairs, and it creates a little dry suit, a little warmth suit around the animal. So that's really an evolutionary kind of leftover that we have. What we see now when we have contraction of this muscle is what we would call goosebumps. Also on this, we have this region right here. This is a sebaceous gland. Sebaceous glands produce an oily substance called sebum. The sebaceous glands release that sebum through holocrine release, and that's when you have the cells filling with the product and then bursting. Right? And we find these on the base of the hair shaft and also throughout the skin. So this is what's going to give our skin the oiliness, which is actually quite important. And I just need to kind of peek around and look here. Over here, if you can see this, I'll move this a little bit closer. This would be a sweat gland, an eccrine sweat gland. These produce what we would call sweat. This is an aqueous product that has salts as well as some uric acid, which are nitrogenous wastes. Right? And that is used in cooling the body down. The lower region over here, this is adipose tissue, so this would be the hypodermis. This region in here where we see all these structures is the dermis, and the very top region here is the epidermis. Now the epidermis has several layers, which are harder to see on the model. Okay, the lowest of the layers would be the stratum basale, or the basal layer, and as we work our way up we would see the different layers of stratum basale, stratum spinosum, stratum granulosum, and then on top the stratum corneum. Right. Now in the scalp the stratum corneum is fairly thin. So let's switch over to the thick skin portion so we can see the difference. So now when we take a look at this, our stratum corneum here is much, much, much thicker. That's what makes us thick skin. And thick skin is what we would see on the soles of our feet and the palms of our hands. We have a much thicker stratum corneum, and that's going to lead to additional protection. What we don't see in thick skin is we don't see any hair. Right? We don't have hair on our palms or on the soles of our feet. So we will see eccrine sweat glands. We do have both dermis and epidermis. Dermis, hypodermis, and epidermis. Okay. What this represents over here looks kind of like a slice of an onion. That is what's called a Pacinian corpuscle. This is a touch receptor. Right? This is a mechanoreceptor that's going to sense pressure. Now the last section we're going to put right towards the middle here. This would be what we would see in the axilla. Right? So the axilla would be the armpit. We'd also see this in the anogenital region. Very similar in organization to the scalp in that we see hair, so we see sebaceous glands. But we also have this type of gland here. Right? This is called an apocrine sweat gland. Now, apocrine sweat glands are different than eccrine sweat glands. Apocrine sweat glands produce a sweat that has a much higher protein and lipid content, and that's one of the reasons why the sweat in the armpits and the anogenital region is different than the sweat that you might see or might have in other areas. So that's just a brief overview of this skin model. The other parts of this lab are fairly straightforward, and they require you to basically look at yourself, right? So to look at the nails, you should use the nails on your own hair, on your own hands, and then to look at your own skin and your own hair.